Good day from Soil Health Academy 2025. I'm Rafiq Islam from Ohio State University. With me, all our presenters, expert presenters are with us, 7 of them. And our media gentleman, Brad Sherman, he's here and he'll cover all these presentations and recording. Okay, I'm Rafiq Islam. I'm the first speaker of this session. And my talk will cover healthy soil and healthy food. I don't know whether it's right or wrong, but that would be my talk. So let's move on and see. So if we say that soil health, food quality, and public health are linked, how we can link our food and health to healthy soil? How we are going to use this loop? So it shows healthy soil is closely related to healthy food, and healthy food supports healthy people or public health. And healthy people or public health, think about how we can improve the healthy soil. So it's all this interconnected, and that makes the system unique to consider how we can improve the soil health. If we move on a little bit more, what it says, recent research on conservation agriculture. That means agriculture, we are talking about conserving, maintaining, improving. Conservation agriculture highlights its effect, how to build healthy soil and producing high protein, nutrient enriched, and phytochemical rich food that will support public health. And how we can do that and how we can define soil health or what management practices can do that. That would be continuous no-till or strip tillage or conservation tillage or reduced tillage. So if we do few years of no-till and then we change into conventional till, it will not work. So we have to have continuous no-till followed by cropping diversity. That means different types of crops, grass, legume, C3, C4 plants, with and without cover crops to get different types of substrate, carbon substrate to carry out the reaction and the processes. Then we talk about precision chemigation. That means how much you have to apply, what we have to apply, when we have to apply, where we have to apply. That is the resource input, fertilization, irrigation, pesticide, herbicides. So that we have to think about optimization. And the fourth one is soil and plant amendments. So liming, gypsum application, organic matter amendments to soil needs to be used to improve the soil health. Same as plant, using aspirin, which is salicylic acid, oxalic acid, and other metabolites to improve the plant's drought resistance capacity and growth and yield. So some of the results will show you. So combination of all these will make or build a healthy soil. And that will support healthy food. And if we have healthy food, then we are going to have healthy people. So you'll find some unique relation how food maintains human health and soil health. Soil health produces healthy food and healthy food supports human health. And some of the indicators or diagnostic indicators tell similar type of comparison or the distinguish between them. pH. So human health, we need to measure blood pH or urine pH, whereas in soil health, we can use water pH or calcium chloride pH, electrolyte, salts, blood pressure. That regulates blood pressure. If you measure blood pressure, we can say, okay, electrolyte level is high. At the same time, human, it would be electrical conductivity and salt. Same thing about air capacity, lung function, where a soil breathe through air capacity, that means pore space, essential nutrient. We do blood tests to find out, but in soil, we see how CEC and base saturation affect soil health. Same about contaminants. We tested blood, urine, and stool. In soil, we measure extractable heavy metals or other compounds. Same as physical status or physical stability or resilience, that is in human health. We are thinking about mobility, but when you go to soil, it's bulk density. If it is bulk density is high, then you are going to have compaction. But if bulk density is low, then you are going to have more pore space and good structure. Versatility, so in human health, it's a bigger. But in soil, with the buffering capacity and productivity, how much they can resist to go into bad or recover, how quickly. Those are the buffering systems. Biotic status, what kind of microbes and how much microbes in human health, that is the gut microbiome or bacterial diversity. Whereas within the soil, we get the similar thing, microbial biome and microbial diversity, earthworm and others. So you can see the similarities, how food can be produced and maintain human health and make a similar kind of thing. So what is a healthy soil and how we can define healthy soil? A healthy soil is nothing but a balanced matrix or balanced system of biodiversity, efficiency, chemical equilibrium, and energy recycling, physical stability, and resilience. When all these biology, chemistry, and physics combine, that's what we get the soil health, the good quality soil or good soil health. But management practices like agricultural system, what kind of tillage or what kind of system we use, that influence the soil biology, soil chemistry, and soil physics of soil health. Because all these science and their interaction, properties and processes influence soil health. How it does? Look at this one, a good soil, and it goes bad, different pieces by primary tillage, and then more small pieces by secondary tillage. You can see all the earthworms, structure, everything gone. What caused that? Conventional system, that means tillage after tillage, recreational tillage, year after year. And that makes the soil, good soil, into bad soil. And it does not take that much time to go into bad. But how we can improve this into good soil or recover the soil from the bad to the good level? So that makes the conservation system to be used, and which will take time to recover, maintain, and improve the soil to the original level or close to the original level. You will never get original level, but close to that. So from this slide, you can see how soil can be bad and what can happen to the soil. And at the same time, if you turn around the system into conservation, how that could be improved over time. So one, conventional goes fast to make the soil bad over time, but conservation takes little bit time. So don't rush to get your soil to be reclaimed. So some of the things I'll show you, the cropping diversity under no-till system. If you look in this slide, you'll find corn, which is C4 plant, rice, C3, soybean, C3, wheat, C3, and brassica, that is radish and legume. These are cover crops. So we have four crops, three crops, corn, soybean, wheat, and three cover crops, legume, cowpea, radish, and rye. And if we grow this thing, what it does, it covers the ground continuously, extended period. The soil will never be exposed to runoff and all this because ground is covered. So the legume and radish will fix nitrogen and recycle nutrients. That will support corn. After corn, we'll have rye as a cover crop. That will control wheat and cover the ground for soybean plantation because soybean doesn't need any extra nitrogen. And after harvesting soybean, the soybean residues with nitrogen will support wheat production with supplemental nitrogen fertilization. So if you use this type of system, continuous cover with different types of cover, with and without cover crops, that makes system more biologically diverse and regenerative. So if we add cover crops, so what it does, it added organic matter, and when this organic matter decomposes, it releases nutrients and accelerates biological diversity and biological activity, which finally provides nutrients and accumulate organic matter. At the same time, it controls disease because of suppression, competition, and allelopathic effect. So the organic matter we accumulate, that supports physical properties of soil. So when all these things happen, finally it improves healthy soil and healthy plants. And with this slide, you can see what types of biological diversity or microorganism in soil can be flourished under different management system to support the system. And biology is the primer actually for the conservation tillage. Without biology, you cannot really recover. So if you look into this one, intensive agriculture versus sustainable agriculture, look at that. The intensive agriculture system is more leaky, soil erosion, nutrient loss, sometimes excessive fertilization because soil is not performing good, bad structure, all these kind of problem. But whereas if you have sustainable ag or conservation, you have optimum moisture level and nutrient supply, good structure and diversity. So these are the system approach to build healthy soil. So if you go more in a summary that all this conservation, resource input versus soil health, how it affects. If we add more fertilizer, more resource, it does not mean that it is going to improve microbial diversity. It will create more pathogenic activities. So that's why we have to always balance a level or optimize the system or optimize the level to get the maximum benefit of soil health. And how we can do that? We have to think where we can get the maximum microbial biodiversity and beneficial organism, carbon sequestration, good crop yield, and other functional activities of soil. So what we got from all these slides about soil health principles, first is minimize soil disturbance. So don't plow your soil again and again. And living roots, that means grow crops continuously with cover crops so ground can be covered. That is the third point, keep the soil covered. When you keep the soil covered, you are going to have living roots and biodiversity and bioefficiency. And then if you have pressure or gas system, if you add animal integration or if you add animal, then you diverse the system with manure and all these. So these are the 5 important principles to maintain improved soil health. So I'll show you some of the examples that what we talk about conservation agriculture can improve the soil health. Look at this slide. soil health, soil quality, percentage-wise, and different management system. CTC, that means conventional tillage corn, no-till corn, and no-till corn soybean, no-till corn wheat, no-till corn soybean wheat, and cover crop. Look at over time, starting from 2000 until 2021, how much soil health has been improved with no-till cropping diversity and cover crops. And first one, if you look at the first part, the conventional till convert into no-till, the effect by tillage, you can see tremendous effect. Immediately put soil into a resting system that improve over time. But when you add soybean, you get a little bit less residues and overall soil health goes down. But when you add wheat as a cover crop or as a crop or cover crop, then you improve the soil health. When you add cover crop, extra cover crop, you improve the soil health more. So here you can see how wheat as a crop and cover crop, winter cover crop, during the winter time it cover the ground, act both to improve the soil health. And this is another example to the left, that is conventional till corn and soybean. Look at the soil. We plowed that again and again, flat structure, and to the right, no-till with corn, soybean, with cover crops. How dark is the soil, undisturbed, and how nice it could look side by side, two plots, two replicated plots. So that's a visual example that how soil can be changed to the good level or to the bad level. It depends on us. So also some effect that how long-term tillage and cropping system improve crop yield. So we think soil health improvement will also increase crop yield. But it may not be the same way. The way soil health improve, crop yield may not be improved the same way. Tillage has minor effect, but soybean has slightly more effect. But in soil health, soybean didn't have effect. And cover crop had strong effect on crop yield because we have converted crop yield, the corn, soybean, and wheat, and normalized because it's three different crops, so normalize and then use the relative crop yield. So it increases, but the way we think, it does not increase that way. So you have to wait or you have to give time to get your optimum yield. So we thought maybe we should correlate that if soil health improvement supports crop yield improvement. So we correlate, we found a little bit curvilinear quadratic response goes up and a little bit down and up. But if you look carefully, this is nothing but linear response. When I tried again, I found almost linear response, but the rate of increase is not that much. So soil health can be increasing faster over time, but yield might take time or lag behind. So if we improve the soil health and soil health improve crop yield, how they are going to affect the food quality? So look at this, the grain protein, how much protein accumulates in the grain. No-till has a little bit higher in both corn and soybean, and plowed system or conventional till, less, way less protein and in both crops. But when you add cover crop, it increases a little bit more. Even if you have plowed soil and you add cover crop, it improves a little bit protein over time. So we analyze the amino acid profiles of soybean, that how this tillage system affect the protein content or the amino acid concentration in the grain of soybeans. And you'll find that leucine, phenylalanine, and tyrosine, they have improved over time. Concentration is higher in no-till system than in plowing system. And more amino acid like other amino acid, arginine, aspartic acid, glutamic acid, glycine, proline, they also improve. So most essential amino acid, few of them improve their concentration because of no-till system compared to the conventional agriculture. So that way we could see how some food quality or grain quality improve over time. So how tillage and cropping system improve nutrient concentration? If you look at this one, you will see most of the nutrients improved over time on all the macronutrients, phosphorus, calcium, magnesium, potassium, sulfur, over time by no-till and no-till with cover crops. And micronutrient, we found iron, manganese, copper, zinc. So they and boron, they increase over time because of no-till system compared to conventional tilled grains with and without cover crops. So based on all this, we calculated food quality index, that how no-till and conventional till affect the food quality. We found no-till almost 50%, I mean, in both corn and soybean improve, but plowed system, the grain of corn and soybean food quality decrease. But when you add cover crop with no-till, it improves little bit more. Even conventional till with cover crop improve little bit more than the conventional till alone. So that shows the tillage is one of the most important no-till system and cover crop. So we also use salicylic acid because salicylic acid is good to protect plants from drought. So it can relieve the plant from drought or improve the drought resistance. And you will find that how they improve the nutrient content, phosphorus, magnesium, sulfur. Mostly these three we found that the grain has, when the plants are treated with dilute solution of salicylic acid, aspirin, it improves the nutrient content, that is phosphorus, magnesium, and sulfur in aspirin treated corn and also in soybean. But when we apply a salicylic acid to see the fatty acid composition, because fatty acid is one of the most important thing for our public health system or for our health, it didn't change that much. It changed, but not that much. So based on all these analysis of soil health and how soil health improved, and it improved crop yield, and then improving crop yield by soil health and all this, how they improve food quality. So when we summarize what we get, conservation farming systems like no-till, cover crops, soil amendments, chemigation work basically integrated to improve soil health. And healthy soil, no doubt, leads to healthier food by increasing protein, vitamins, and other phytochemicals and nutrients. Among the conservation system, no-till with and without cover crop boost protein, essential amino acids, and nutrients in corn and soybean, and improving food quality. And salicylic acid helps plant or crops to tolerate drought and increases their nutrient concentration and phytochemical content. So based on this, our One Health vision is healthy soil. Soil we get, miracle, we get it as granted. And then healthy food, look at these tiny blueberries, what nutrients and all these in these tiny blueberries, and soil and public health or human health. So One Health, soil health, healthy food, and soil health, all are interconnected to support ourselves and the ecosystem services in our world. So that's all I have. Thank you. If you have questions, I'll be glad to answer.
Thanks, Rafiq. Yeah, we want to try to stay on schedule, so we want to get Jason on here to give his presentation. But Rafiq, there were a couple questions that came in in the Q&A, and maybe you can type some answers out to those as we start the next presentation.
Thank you. Thank you.
All right, Jason.
All right. Good morning and good evening, everybody. It's great to see so many folks online and participating in our Soil Health Academy. So this presentation is going to focus on soil biology. And so I'm going to focus on a number of different topics related to that. Before I get started, I just want to draw your attention to a group of different presentations that are available related to soil health, and I'll put those in the chat a little bit later on. But it's something that we've been working on with the USDA and RCS, and you can see them identified here on this title slide as well. So to get started, basically, we know that there are a lot of benefits that the soil provides, and I've got a number of those identified here. And the question here is, which of these soil functions are influenced by soil organisms, whether they're macro or microorganisms. And the answer really is that all of these different types of functions and benefits are influenced by soil organisms in one way or another. And I'll talk about some of these different soil organisms and how they play a role in soil health. So just to kind of start out, we see a lot of this information about the organisms that are in the soil. There's a lot of them. But a lot of times they're very small. They're going to be microscopic, but they definitely add up. And so this chart gives a good depiction of that. And you can see the relative proportions of different types of organisms. If you look at the top, essentially 1 hectare of soil or 2.5 acres is going to contain the weight of 20 cows when you add up all of the different organisms that are beneath the soil. And so that's a pretty significant load, and it really goes towards helping to provide the soil health benefits that we've already been talking about. And so that's just kind of a snapshot related to how important they can be. Now, there's some folks with USDA, Natural Resources Conservation Service, who have kind of grouped these individuals, these organisms, into 3 functional groups, and you see those here. We've got some soil ecosystem engineers, then we've got biological regulators, and we've got chemical engineers or microbial decomposers. So I'll talk a little bit about each of those. So the chemical engineers, you can see some examples of those. They're essentially the ones that are contributing to the buildup of organic matter as well as aggregates. And so the aggregates are essentially the binding of soil into material. Anytime you dig in the soil, you're going to see these kind of aggregates come out. And those are naturally formed in a number of different ways, but a lot of these organisms are important in that development. And then we've got biological regulators, which are essentially keeping our microbial populations in check. And so that's really important so that there's no one group of microorganisms or organisms that kind of take over, because that's going to be a detriment to the soil and soil health properties. And so that's an important group that plays a role in the soil. And then lastly, we've got these ecosystem engineers that are building the pore spaces, the aggregates as well, and they're distributing soil particles and organic matter throughout the soil. And one that you think of off the top of your head is going to be earthworms. They're kind of the natural tillage systems that we've got in the soil. They're moving materials around, whether it's from deep in the soil up to the surface or vice versa. And they really play an important role in ensuring that that litter material is recycled and help contribute to aggregate formation and all that good stuff. So when we're talking about good soil health from a biological standpoint, we really want to make sure that we are maintaining good populations of these soil organisms. We want to make sure they have what they need. So for us humans, that's going to be food, water, and shelter. And for these microbes and macrobes, it's essentially a similar situation. So I've got a list of different things that are important for these organisms, and essentially we're talking about kind of the Goldilocks situation where we don't want anything too extreme. We don't want it too hot, too cold, too acidic, too basic. And so we want to keep it in that happy medium there that everything tends to like in the soil. And so there's a number of different places in the soil where you're going to see these biological hotspots. And I've got 5 that are identified here. We've got the litter layer, we've got earthworm channels as well as root channels, we've got pore spaces, we've got aggregate surfaces, and then we also have the root zone. And I'll talk a little bit about each of these as I go through the slides. The first one is the litter layer. So the litter layer, of course, is going to be important for protecting the soil. It also keeps the soil in place so we don't have erosion. It helps to maintain temperature and moisture, which was already discussed before. It also serves as a good carbon source for soil organisms. So they're going to be taking this litter material and using it as a food source. And so they will also, in that process, they will also be turning some of that carbon into different forms that can be useful in soil health, which I'll talk about a little bit later. So these are some of the different organisms involved in that litter layer. And again, you're going to find a lot of organisms in this area because it's got a food source, and that's something that these folks are really interested in. Now, when it comes to the pore spaces, these are referred to here as the lungs and circulatory system of the soil. So in this case, it allows for gas exchange and airflow. It also allows for water movement. And so those things are really important. In the lower left-hand corner, you can see the chart there. Ideally, we like to have a soil that's made up about half of the volume space is going to be solid material, and then the other half is going to be space. And that space is going to be represented by either air in various forms of gases, or it's going to be water. And that's kind of our ideal situation. We don't want things too dry or too wet because plant roots as well as organisms in the soil prefer a good balance of that. And so these are also good biological highways because there's a transport of gas and water. And so this is a good habitat for our biology in the soil to kind of hang out. The water provides nutrients and it also allows for some transportation as well. Now, on the right-hand side, I've got some different diagrams that kind of show the difference between a well-structured or a poorly structured soil. We like to have some good pore spaces. That's basically the idea here. And in the top right-hand portion of the slide, you can see some microscopic images of the soil. On that left-hand side here, we've got a situation where there's a lot of good pore space. You can see a lot of space in there. On the right-hand side, this is more of your example of a tilled soil. The aggregates are broken up. We've got a lot of the pore spaces plugged up, and that's not really going to be great for water movement or airflow or anything like that. Now, aggregate surfaces, you can see here, this is a typical image of looking at aggregate stability. And so essentially, we're able to, if we've got good aggregation, when when it comes into contact with water, it's going to hold its shape, it's going to hold its form essentially, and that's going to be great for preventing any sort of soil erosion. But these aggregate surfaces are important because they also are a good habitat for microbial populations. And those aggregates are also going to protect organic matter from further decomposition by microbes, because inside of those aggregates, it's going to be a little bit more difficult for some of those microbes to access that material. And so in that way, it's going to be protected and in some cases sequestered. And so the aggregates themselves are developed by some of these microbial glues, if you will, as well as fungal hyphae or plant roots, and that's going to be really important in that aggregate development. So this is just an example of that. So you can see the roots, of course, play a big role. They can create a mesh around the soil and create these aggregates naturally. Anybody who's dug around in the soil is going to see that soil kind of attached to those plant roots. Then we also have some of these bacterial filaments that are created by microbes, and they are sticky and they can hold particles together. And then we've got the hyphae of fungi, and those can also wrap around soil particles similar to like a root system, and keep those soils together and bound together in those forms. So we've also got earthworm and root channels. These are another good place for microbes to be found, as well as macrobes. There is some food there, particularly with the root channels. We have a lot of root turnover. So there are roots that die, and then there are new roots that are formed. And those dead roots are a food source. They're a carbon source, and so they can become decomposed in the soil as well. And of course, that food source is going to invite additional microbes to come and hang out. And so there's a number of examples there of some additional benefits of these root channels in these areas. So, with respect to tillage, it definitely has an impact on microbial activity. If you think about what I mentioned before, where these aggregates are kind of broken up, and it makes the organic matter that's inside them more available and more exposed to microbial activity. So, under a conventional tillage system, after you till, you're going to see this huge increase in microbial activity, and then it's going to die off. That indicates essentially it's similar to the populations. The populations are going to increase significantly because there's a huge food source, and they're going to really taper off after that. And that's not really what we want. We want them to be there all the time, essentially, but we don't want the populations to get out of hand. So in a no-till system, this is more like what we see. We see a very gradual increase, and nothing really spikes throughout the growing season, and then it tapers off as temperatures change. And so that's really a benefit, because we maintain similar microbial populations within the soil, and that's going to really be a benefit to soil health activity. And so with the root zone, again, we've got a food source here. So roots are going to provide some organic acids and other types of carbon sources. They are exuded from the roots themselves. And so that's going to attract microbes as well as other benefits to the soil and to these specific areas. And so I like this diagram. You see this a lot in different areas, but this really shows a good example of the diversity that's out there. And the diversity is really something that we want to have, particularly when we're talking about microbial populations. And so in this case, we're just looking at the root architecture itself, but it's a good example of the differences in plants. So each plant is going to exude different types of carbon sources, different types of organic acids or other types of carbon materials that the microbes are going to use as food. And so each plant and the carbon source the carbon that it releases is going to attract certain types of microbes that can break that carbon source down. So microbes have different enzymes that they use to break down these carbon sources. And so that's why you want to have a diversity of plants, because they're going to release a diversity of these carbon sources, which is going to attract a diversity of microbes. And so having that diversity is really useful because they can break down and they can do different things, and so that's a benefit having that throughout the soil and improving the structure of the soil and the soil health principles. And so this is just an example of this. As we get more species of different types of plants in a system, we're going to see an increase in the microbial biomass. And generally, it's not just an increase in the microbial biomass, but an increase in the different types of microbes that are out there. And so again, it's that diversity of different carbon that's released from the root systems that are attracting these populations. And so the soil organisms themselves, I mentioned this early on, is they are also releasing materials that can bind the soil. And so these soil proteins or polysaccharides act as a glue that can stick particles together and create these soil aggregates. The microbes are also important in a lot of our nutrient cycling. And so specifically with nitrogen and phosphorus, these types of specifically bacteria are going to play an active role in some of the different types of nitrogen cycling that we see here. So each one of these arrows essentially is going to have some kind of a microbe that's involved. With nitrogen fixation, we've got rhizobium that are involved with interacting in legumes. And so that's an important role that they also play, making some of these nitrogen species more available for plants to take up, and releasing some of the organic nitrogen that's present in our organic matter, so that plant roots have access to that. And then also with phosphorus, it's a similar situation. So in this case, we've got these mycorrhizal fungi that are going to interact with the plant roots. And so the plants, it's a perfect symbiosis where the plants are providing carbon to those fungi, and then the fungi, the hyphae of those fungi are very small, smaller than plant roots, and so they're going to be able to access certain areas that plant roots can't. And then they also have certain enzymes that can help to release some of these more recalcitrant forms of phosphorus. And so that's what the plant gets in return. It gets the phosphorus, and the fungi get the carbon from the photosynthesis of the plant. And so, how do we support that biodiversity? And this was alluded to in the previous presentation, but these soil health principles, we want to protect and we want to feed these micro and macro populations of soil organisms. And so there are kind of these four principles that we look at in being able to do this. And so there are certain agricultural management practices that are associated with these soil health principles, and you can see them identified here. We've talked about tillage, we've talked about cover crops. The cover crops and the crop rotations are really important because, again, it provides that diversity of different plants and different organic material that promote a diversity of microbial and macro populations. Perennial crops are great because you're reducing tillage. Organic fertilizers are great because not only do they provide nutrients, but they also provide that organic material. Any kind of excess fertilizer or excess herbicide or pesticide use is going to be of issue. When we're talking about pesticides, generally they're not targeted to affect the organisms that you want to get rid of. They're going to affect everything. And so that's going to reduce the good populations, the populations of good microbes that are there as well. So there are some other things we can engage in, some cultural practices, as well as some integrated pest management practices that can help to to mitigate some of those issues that we see from broad spectrum pesticide applications. And so with that, I'm going to conclude, but I want to thank folks for tuning in, and I'll be happy to answer any questions that folks put in that Q&A spot there. Thanks.
All right, thanks a lot, Jason. And yeah, we have a few questions. I see that Rafiq is working on one right now, and he and the others will certainly get to them as they can. And I think it's time for Sean. I guess you're up next, Sean Lucas from University of Vermont.
All right, I'll share my presentation and get going here. Thanks, everybody. And hopefully that's coming through okay.
Great.
I'm going to talk a little bit about soil health assessment. I saw some questions in the chat regarding, what tests are we using to assess soil health and make interpretations. That's kind of going to be the crux of what I'm talking about. I am at University of Vermont. I am extension agronomy, assistant professor of extension agronomy here. Most recently, prior to this position, I was at Kentucky State University, where I was assistant professor of organic ag. But I've been looking at soil health for the better part of about 25, 26 years. And so it's neat to see it become mainstream these days. We've talked a lot about some of these basic soil health facts. I'm going to kind of skim through this because I want to be respectful of our time. 20 years ago, we were talking about soil quality, and that was really the ability of a soil to carry out certain ecosystem functions. And that might be, is it building shelter, or is it growing crops, or making pottery, right? So the function of the soil, what you're trying to use it for, helps determine its quality. Fast forward a decade or so, and you see soil health really become more prominent in the literature and in the discussions between technical service providers and researchers and farmers. And that's because, we really are thinking a lot about how can we improve our cropping systems? How can we make our cropping systems sustainable? And we've really come to the understanding that biology, as Jason so eloquently shared with us, really plays a big role in soil health. So as this importance of soil biology has taken precedent, we've really kind of started thinking about, instead of just this quality idea, let's call it soil health and really recognize that importance of biology. You'll see these terms used interchangeably. It's less so now. I think people have really kind of they understand the distinction. But whether we're talking about soil quality or soil health, people have been trying to assess it, quantify it for the better part of the last 2.5 to 3 decades. And we want to do this because it can help us make management decisions. We want to understand what the best management practices are, particularly for someone like me who is in extension now. I want to be able to go out and share with stakeholders and farmers data-driven information that can help them make good decisions in their operations. I think we've had a little bit of primer on soil organic matter already, but this is really the key in my mind and in a lot of people's minds. Soil organic matter as affected by management practices. That's the key determinant of soil health. It's related to nutrient cycling. It's related to soil structure, which really dictates air movement, water holding capacity, water infiltration. And it's the food for the biology, which is where we're, doing all the biology is facilitating all of the nutrient cycling that feeds our crops. So soil organic matter is a really key part of this, and it's manageable. We can influence soil organic matter content. That's pretty well established these days. It responds to good or bad management. I've got a couple examples on here that will be deleterious or cause soil organic matter to degrade. So tillage, especially the type of moldboard tillage where you're getting that full inversion that you see in the photo here in the upper right. Erosion, you're taking the best topsoil off, and it ends up somewhere where you don't want it, and then maybe you're farming in subsoil, which is not necessarily the best condition to be farming in. And then monoculture crops, that lack of biodiversity, you're having the same nutrient profile consistently extracted from the soil by that particular crop. And it's really driving sort of deleterious changes in soil organic matter. And just, since we're talking about soil testing, typically speaking, the rule of thumb, if you're sending a soil sample in, and they're doing loss on ignition, soil carbon testing, soil carbon is roughly, the rule of thumb is soil organic matter is roughly 58% carbon. So on your soil test report, typically you'll get a soil carbon score and a soil organic matter percentage. Really, they're just multiplying the soil carbon by 1.72 to get that soil organic matter. As I mentioned, it can be changed, right? Soil health can change depending on how we're managing the soil. So if we're doing things like a lot of tillage or monoculture cropping without using cover crops, you might see a deterioration. If you're adding amendments, organic amendments like manure, we use a lot of manure here in Vermont, cover crops, no-till, you're going to go the other way, and your organic matter is going to accumulate until you reach a new equilibrium. And there's a stabilization process in there as well, where as you are shifting management practices, things will change, but then they'll ultimately kind of stabilize and reach a natural equilibrium. And I want to put this slide in here, I want to highlight this because it kind of relates to the things I'm going to talk about after. A lot of the thought that I put into this was about interpretation of soil tests, soil health tests. And it's really important for farmers to understand their own system for them to make sense of soil health tests, whether they're coming from commercial labs or university labs, which I'll talk about here in a minute. So farmers have kind of had a good understanding, if they're good land stewards and good managers, they have an understanding of really how their soil is functioning. They may not even need to necessarily go and do a bunch of testing if they've been understanding their system for a long time. How does that soil feel? Does it crumble into aggregates? Well-formed aggregates, or is it hard clods and kind of crusty when you break it up in your hand? How does it smell? The aroma? There are compounds produced by some of the soil microorganisms that kind of give it a fresh, earthy smell. That's usually a good sign. Or, if you smell fermenting byproducts, that might mean there's lack of porosity, lack of oxygen in there. How's it look? I have a picture here. At the bottom left, those are my former advisor's hands at University of Maryland. That was Ray Weil. Those two soils are from adjacent fields. One was no-till, one was long-term till, and they're the same soil series. That's the effective management. And so the soil on the right, if I'm making a interpretation just visually, looks a little healthier. You can see better aggregation. It's got a darker color. The soil on the left, you might want to start implementing some better soil management practices on that. And Ray Weil and Joel Groover, when I was at Maryland, they were working on this project. They were looking at farmer perception of soil health. And the crux of what they found was that the strongest relationships with farmer perception of soil health were correlated with soil carbon parameters. So farmers have a good sense, and that benefits them when they're working with technical service providers or making interpretations on soil health testing results. All right, so we can assess this stuff. We can test it and try to get a determination of whether a soil is a healthy soil or you might need to do some things to make it healthier. Well, what do you want to look at? You want to look at how the soil performs all of its functions. You can't just look at crop yield. You can't just look at water quality. You have to look at the big picture and a lot of different indicators, and that'll give you a sense of how well that soil is functioning. And that's really what a lot of the soil testing labs that are offering soil health assessments are trying to approximate. They're trying to get multiple readings and give you a soil health score. And again, you really want to look at how all the functions are in a snapshot, but then over time, we'll see this as I talk here, that these soil health tests, they're all kind of relative to one another. So you have to really see how your soil was functioning initially when you first test it, and then see how it's changing in time as you change management practices. The best soil health indicators are ones that change with management over a few years. That doesn't mean these other indicators or tests that you can do, like pH or available phosphorus and potassium, those can change pretty rapidly, and we can influence those pretty rapidly by adding fertilizer, by adding lime, right? It's a little harder to change texture. That's a more permanent property, but that you really have to have an understanding of that to make an assessment of soil health. But the ones that are going to give you your management response are these indicators like soil aggregation, microbial biomass, permanganate oxidizable carbon, which I'm calling active carbon here, and a little bit longer term total soil organic carbon. We really are looking at the big picture, right? This was one of the coolest things when I started studying soil science. I was like, wow, this is pretty cool stuff. You can't just be a pure scientist. You've got to understand biology, chemistry, physics. Well, when you're looking at soil health, you're looking at biological properties, chemical properties, physical properties. Some of the biological properties we look at are respiration, microbial biomass, soil biodiversity, different carbon pools, physical, soil structure, water retention, porosity, texture. And then chemical, things like nutrient holding. Certainly pH is a big driver of how well soil is going to function. Cation exchange capacity plays into soil's ability to filter out pollutants and also plays into nutrient holding. And then you can have some things that are, kind of both. If you look at biochemical or biophysical, rather, a good example is, soil structure is a physical property, but soil structure is influenced by the biology in the system. And so that's really actually kind of the aggregation is a biophysical parameter that we're interested in. So when you get into soil testing to determine soil health, which has really come on over the last decade and a half, a lot of labs, I shouldn't say a lot, there's kind of limited options, but you got choices. You got commercial labs, and then you've got a few university labs. If you're interested in this, go into it eyes wide open, knowing that this is more expensive than sending your sample off for a nutrient profile and getting recommendations, and it's less well established. We'll talk about that in a minute. But we kind of know how to interpret soil fertility testing. You get a phosphorus reading, you're going to either be low, medium, optimal, or excessive, right? And then you can get fertilizer recommendations based on that score. It's a little more nuanced and a little less clear sometimes with soil health assessment. But I will kind of give you an overview of some of the things that are out there. I'm not recommending any particular lab. I'm just giving you an overview. When I was in Kentucky, a lot of the farmers used Waters Ag labs. They had a couple of different stations in Kentucky where you could send your samples. They're actually based in Georgia, and they serve other states beyond Kentucky. But this was a commercial lab. They offer a sort of a basic soil test that gives you a complete nutrient analysis. They look at nitrate, nitrogen, ammonium, nitrogen, Solvita CO2 burst test, which is basically a respiration test. The Solvita SLAN, which gives you a available nitrogen assessment. Permanganate oxidizable carbon, which is that active carbon. They're doing an aggregate stability measurement. They're looking at total organic matter. Another commercial operation is Ward Laboratories. Again, they're sort of a national outfit. They actually do PLFA, phospholipid fatty acids, which will give you an assessment of microbial community. The Haney test, they do enzymes, which was kind of neat. I don't see a whole lot of people doing enzymes, but if you're interested in that, and you have to be interested in it with an understanding of how to interpret it, Ward does give an enzyme set of enzyme readings. They're doing active carbon, total carbon, aggregate stability. They'll give you a water holding capacity, total nutrient digest. So they're giving you some of that standard soil testing information for fertility that would go with the soil health. And they're doing autoclave citrate extractable nitrogen, which is also a sort of an available nitrogen test. I did look at their page and I thought this was kind of interesting. You know, they had this quote on their page, widely accepted by land-grant university correlations and calibrations. Just the way that's worded kind of, you know, it seems like it's trying to give it credence. Correlations and calibrations, they're tools, but they don't accept things, right? So I just thought that was interesting. There are several soil health labs associated with universities. I don't know that this is an exhaustive list, but I wasn't able to find a whole lot more beyond the 5 options here. And if anybody knows more, drop them in the chat. I'd be real interested to learn about them. Cornell, I'm up in the Northeast now, and Cornell is actually probably the most well-known university associated soil health lab. We've recently started one here at the University of Vermont. My colleagues Joshua Faulkner, Heather Darby, and Deb Nair actually got this up and started to get this up and running before I came on board at University of Vermont, and now I'm working with them on this. But the Soil Health Research and Extension Center, SHREC, and we're working to get that up and running. There's a soil health laboratory at Oregon State, and then SHAC is at the University of Missouri Soil Health Assessment Center. University of Maine will give you a soil health add-on option if you submit a sample for basic soil testing. And I don't know that I have time to go through these, but you can see some of the tests are similar across the university labs. Cornell is interesting that they'll give you penetrometer, that you can take penetrometer readings, and they'll give you sort of subsurface hardness interpretation, compaction interpretation. The neat thing about Cornell and University of Vermont is that they're using regional database. So they've collected information on regional soils, and they're basing their interpretation of the results on those regional databases. Part of the reason that we established a lab here at University of Vermont was because even though New York's right next door, we had farmers here in Vermont saying, well, I'm getting these soil tests from Cornell, and they're saying I've got high soil quality, but I don't think, or high soil health, but I don't think my soil is functioning as well as it could have. I'm not getting the yields I might like to see. And so we established a lab here using Vermont soils. Eventually the goal was to perhaps go beyond Vermont, but right now we're mostly servicing Vermont stakeholders. Again, similar suites of tests at Oregon State and University of Missouri. All right, so interpretation. There are some caveats when we're thinking about soil health interpretation. Determining what is good in a specific soil type. In other words, you've got a sandy soil or a clay soil in the same region. What's good in both those, what's good soil health in both those soils might be quite different. And so we really need to interpret our tests with an understanding of what soil we're working with, the region that that soil is coming from. And again, I pointed that out. One of the things I like about the Cornell assessment and what we're doing here at University of Vermont is that it's based on regional database. We're actually, the lab here at University of Vermont is just opened up this year. We had a big announcement earlier in the spring, and we're in the process of building that database now, so that we can make Vermont specific recommendations. I touched on this earlier, but these tests are a snapshot. So, if you send in a soil sample and you say, I want soil health assessment, and you get back this interpretation, the first one might not really give you a whole lot of information. It's when you take that first one and then start looking at it over time as you're adjusting your management practices. And so I think that's right now the best way to think about soil health assessment. There's not a magic number that says, oh, it's a score of 75. That's great. It's over time and monitoring. We know that soil biology can vary widely across the field, and a lot of these parameters that we are interested in measuring are soil biology related. So you really have to think about how you're sampling, and you want to, just like if you're taking a fertility test, you don't want to hit hot spots where the manure pile was stored last year, right? You want to, or you don't want to sample where you see earthworm middens exclusively, right? You want to get a smattering of everything that's going on in a particular field. And so you've got to use that same kind of sense for soil sampling, so that you get a representative test. You might not see changes immediately to management practices. Test sensitivity, permanganate, I've been working with permanganate for the better part of 26 years. 2-3 years, you might start seeing changes, but really over 4 or 5 years is when you really start seeing that sensitivity to management, consistent management being evident. And subtle changes, they may not be as evident, or they may take longer. And I don't know, my panel is over on top of my table here when I see the panel of panelists. So you can't see the column on the left or on the far right, but in this table, it's got no-till and cover crop, no-till, no cover crop, and then there was tillage, no cover crop. And the point with this table is, if we could see, I can't see the tillage, no cover crop column, but there's asterisks next to all these numbers: the quality score, the aggregate stability, the organic matter, the active carbon, soil respiration. There's might be one that doesn't have an asterisk, but most of them are indicating a statistically significant difference compared to the other two columns. But when you look at no-till and cover crop versus no-till, no cover crop, even though the no-till and cover crop numbers are higher, those aren't statistically higher in this particular study by Zuber and Kladivko. So that gets to this point that subtle management changes may not necessarily be as evident. I've got 1-2 more slides here, and I just want to make this point. Again, knowledge of your farm and knowledge of your system as you interpret this or work with your technical service provider to interpret this is important. This is a Cornell, and I'm not knocking Cornell on this. I really like what they do. You can see here there is a lot of indicators in green: available water capacity 91, organic matter 99, active carbon 94. And then you've got a few that are sort of in yellow or orange, which is sort of caution or medium high score in terms of soil health. But if you look at the overall quality score, 77, it's high. It's considered high. And that 94, the nice thing about Cornell is they'll give you a 10-page readout in their soil report. And some of the commercial labs aren't necessarily as robust in like telling you what they do to get your score. Cornell's very good about that. But if you look at the active carbon readout in the Cornell interpretation, it tells you, well, this is a high number. Your management is ideal and you want to maintain this. Well, if you look at the pictures on the right, that's the field. So we're getting this high score, and this farmer on other fields in Vermont, he's in Addison, Vermont. He has won awards for his corn yields, but he will tell you that this field is probably not one of his best fields. Even though it's scoring high in quality, you can see crusting. You could see some water logging, 6 inches down. I was actually taking a cache test this spring at this farm, and when I dug the hole to collect the cache sample, the hole filled with water. All right, so maybe you need some other things like tile drainage there. But to just read the score without the knowledge of what's going on in the field, it can make interpretation difficult. So final takeaways here. We're looking at multiple facets of soil properties, different indicators. These are not as established as soil fertility tests. I pointed that out earlier. There's a little more nuance and uncertainty that goes into interpreting soil health tests. I like to look at them as a snapshot and then see how things are performing in subsequent snapshots. Again, and I can't emphasize this enough, what might be a healthy soil in one region might not necessarily, or one soil type might be qualified as healthy, might not necessarily qualify as healthy in another soil in another region. Soil health test interpretation needs to be informed by regional factors and producer knowledge of the system. The distinction between commercial labs, the prices, they're both more expensive. Whether you're going with a university lab or a commercial lab, it's going to be more expensive than a basic soil test, or your standard soil test where you're getting nutrients and cation exchange capacity, et cetera, et cetera. Commercial labs might be a little faster, but at least from what I'm seeing, the university labs seem to give more regionally appropriate interpretation. So, I would say do your homework if you're thinking about using soil test labs. And I will say we have a lot of international participants here. This is geared more towards a U.S. Set of tests. I'm not well versed with what's going on overseas yet, but I do think that one of the things I'd like to emphasize is that we probably need more labs tailored to specific regions. I highlighted 2 examples of commercial labs. They're kind of national operations, and then we have some regional based university labs. But I think we need to get more labs tailored to specific regions. And I don't know where I am on time, but I probably have to take questions in the chat.
You're pretty close. Thank you, Sean. And our panelists have been doing an excellent job keeping up with the Q&A. We went through about 13 questions so far. None are in the queue right now, but if anybody has a question for Sean on his presentation or for anybody else, feel free to drop them in there. But we're going to keep on schedule. Dr. Barden from our friends at Tennessee State, he's up next, and I'll turn it over to you.
Okay. My presentation up.
Yep, we see it.
Okay. Thank you very much. So my name is Sugata Barden. I'm an associate professor of soil health at Tennessee State University. And so far, you have had talks that talked about soil biology and how soil health relates to food and importance of soil health in terms of food production in general. This talk, I will focus mainly on management practices and keep it broad, and I will suggest a more systems approach on how we can look at a systems approach of managing these practices and try to manipulate the soil health. So I'll start with this slide, and this is not my slide. I will like to give credit to my collaborator, Dr. Walton Sumner. He used it in one of his presentation, and I really liked it. So what is the link between these ancient civilizations? Because we don't have a live audience, I will move on to the answer. Is that these are all remnants of mighty empires, many of whose downfalls was caused by degraded soils, intentional or not, and are now classified as developing countries. So the bottom line is ruined land, hard scores of generations. So these are at one point used to rule the world throughout history. So I won't continue on this slide for a long time, but why soil health matters. I mean, the previous presenters kind of alluded to most of these things, but to briefly summarize, soil is the foundation of all terrestrial life, including food security, water quality, and resilience to climate change. So healthy soil is important for nutrient cycling. It supports diverse microbial life. It is important for recycling of nutrients. Healthy soil is important to regulate water flow, and also soil sequesters most amount of carbon on the planet. However, it is also important that globally about 50% of the soil that is under agricultural production is currently classified as degraded, according to FAO, which further emphasize the need that we have to look at soil health very urgently, and we have to take steps by which we can maintain soil health so that we can maintain the productivity of soil for generations to come. Now, the title of my presentation is Management Practices and Soil Health. So what are the management practices we are going to talk about? So management practices are techniques and strategies that are used by farmers and agronomists to improve crop production, overall farm sustainability. So these practices are grounded in agronomic principles, the science of soil management and crop production. So creating a better seed bed and providing the ideal condition for the plant to grow. So if we look at definition of soil health, there are many definitions of soil health. I like this one. I mean, probably one of the first definition was given by John Doran, but I like this one better. Or I should not say better, but this encompasses most of the things. So soil health is the capacity of soil to function as a living system with ecosystem services that support agricultural production, biodiversity, and environmental quality, because we want to have all of these attributes for a healthy, sustainable ecosystem. So let's look at some of these management practices that we are talking about. So these include tillage, nutrient management, crop rotation, There are many others that are also included in management practices, such as irrigation, water management, pest and weed management. But we are not going to be able to discuss all of them, but we will look at some of them in little more details. So let's look at tillage and soil health. And some of the previous presenters have already talked about the importance of tillage and how tillage impacts soil health. So here is some of the pictures that shows how tillage can impact soil health through erosion. So repeated tillage can destroy the soil structure. So where you do not have aggregation, so through rain erosion, water erosion, you can have gully formation, and you have a situation where you have degradation of your landscape. Because of these situations, we have had alternative management practices. So again, I mean, we are trying to manipulate these management practices to improve soil health. So we developed conservation tillage and no tillage systems, which preserve structure, enhance biopores, and also support biology, which Jason mentioned, earthworm activity. So this picture on the extreme right shows earthworm hole. This is a study done at Coshocton in Ohio, where they tracked the earthworm holes by smoking through the tile lines. And this one was in a no-till field, which almost reached to the tile line up to 3 centimeters of the tile line. So we have to also look at the benefits of everything and also the disadvantages. So we know no-till systems are beneficial in terms of preservation of soil structure, reduction in erosion. It enhances water infiltration, but it also can be a problem in terms of in situations where you apply manures, for example. So I was a graduate student at the time, and this was a study done during my time. I was not part of this study, but I did help extensively, where they did a study by excavating 30 by 30 by 30 soil blocks, intact soil blocks from 6 tilled and no-tilled soils, and then they applied manure, and then they subjected these blocks to rainfall, and it was observed that the no-till fields caused transport of many of these pathogens through the tile lines. So we have to adjust our management practices so that we are aware of all kinds of impacts. So again, the goal is to manage these systems so that we can find the best management practices so that we can create the ideal environment. Next, we'll talk about cover cropping. Again, you have heard about cover cropping, how these are beneficial in terms of creating a ground cover, helping with nutrition, and also suppressing weeds. We had a project. It was a USDA OREI funded project. It was looking at whether having a long-term cover crop project, organic project would allow enhanced disease suppression. So the goal was to see if long-term organic management practice using cover cropping will impart enhanced disease suppression capacities to the soil. So what we used was, and this study was conducted in partnership with Dr. Chang-Ho Lin at the University of Missouri Center for Agroforestry. And we used a metabolomics approach, and we profiled over 1,500 distinct molecules from the soil. And what we saw was that the treatments under cover crop showed a more diversity of metabolites. And many of these metabolites had antimicrobial, antifungal, and antibacterial properties compared to the control and no cover crop treatments. So this is the heat map that was generated. So these are color coded. So the first two columns, so the red and green on the extreme left, so these are our control. The next two, the two shades of blue is our treatment with no cover crop. The next set of three is our cover crops, first year cover crop treatments, bare tilled, flail mode, and rotary mode. And the last set of three are 5-year cover crop treatments. And the heat map shows the blue is less concentration of these metabolites, and you can see on the right are the different metabolites, and red means high concentration of these metabolites. And you will see that with the set of 5-year cover crop treatments, there is a high concentration of many of these metabolites. And the theory is that these are going to impart an inherent disease resistance. The work is currently ongoing. We are going to do bioassay against some known pathogens, and we will find out what the results are. Next up is crop rotation. Again, crop rotation, again, it was discussed in the first talk, and I'm not going to spend too much time here, but the idea is it reduces pest and pathogen pressure and helps with a healthier soil environment. And lastly, integrated nutrient management, which also helps create by incorporating compost, biochar, and organic amendments. We can create a healthier soil environment, which can promote better productivity and create a better environment for the crops. Now, these are some of the management practices that I will discuss, but let's look at what is the current state of farmlands in the US. Like Sean mentioned, this talk is mainly targeted for the US, although there are a lot of audience from overseas. But if we look at the overall farmland in the US, 40% of the US farmland is classified degraded. About 12 million hectares are lost to soil degradation and desertification from 85 to 2015, according to USDA. Studies estimate that soils in the parts of US have lost about 30 to 60% of their original soil organic matter content since the beginning of widespread farming. And this is a rough calculation. So this shows the decline in organic matter. Somewhere and this is average. So we started from about 5%. So right now we are about 1.6% organic matter. And even now we are losing a lot of soil, about 1.7 billion tons of soil annually due to erosion, which exceeds the rate of natural soil formation. Although we don't see this type of catastrophic dust bowl clouds anymore, this is still a lot of erosion that is happening. So what does that mean? If we look at overall productivity overall in the world, we have seen, I mean, this is major crops. This is from one of our papers from the Green Revolution from the 1960s to the 90s. For the first 30 years, there was increase in productivity, but in the next 20 years, there has been a decline in productivity. And the population of the world is going to increase. So where does it lead us? So we need to think about soil health and how we increase productivity. And that brings us to this, again, looking at the broad picture. So we have all these resources, water resources, land resources, plants, human settlement. So everything is interacting with each other. And on top of that, we have the climate, human activities. And when everything interacts and we have a favorable outcome, we have sustainability and resilience. When it is unfavorable, we have degradation and vulnerability. So we have to have a situation where we are in that favorable column and we are heading towards that green corridor. So how do we do that? Again, it goes back to the management practices. So we do that by increasing biodiversity. Some of the speakers have talked about how a diverse system is more structurally sound. So we increase biodiversity, we add organic fertilizer, we add crop rotation, we add cover, and we achieve all of these. We lay the foundation for a healthy soil. But like I said, we are in a situation which is not sustainable. We have been talking about sustainable agriculture before we talked about regenerative agriculture. So we should have talked about regenerative agriculture first, and then we should have talked about sustainable agriculture, because we degraded it too much, and we should not sustain what is degraded. So we first regenerate, and when it is in a state of equilibrium, then we can sustain it there. So we have to regenerate the degraded landscape first. So how do we do that? So we have to look at a systems approach. So combining practices. So a holistic systems approach that integrates multiple practices for cumulative benefits. So combining trees, animals, organic amendments, this creates a synergistic effects on soil health. I had been working on agroforestry systems, and when industrial agriculture started, at least in these countries, about 200 years or 150 years ago, most of the trees were removed from the landscape. And the graph I showed, the decline in organic carbon, that all happened in the last 150 to 200 years. And we now know that was not the best decision that was done. And I understand that we have to maintain and increase productivity, and we industrialized agriculture. We have big machinery. We have to move through this landscape. We invented fertilizer, pesticides, but nature has its own way of operating. So we have to, instead of fighting, we have to make it so that we can merge things with nature and make it so that we can work synergistically with nature and at the same time increase what we are trying to do. So that gives us the opportunity to reintroduce trees back into the landscape. So agroforestry gives us that opportunity, which will allow us to improve soil health through incorporation of trees. So let me introduce the different agroforestry systems. So alley cropping, silvopasture, forest farming are the three agroforestry systems, and riparian buffers and windbreaks. These are more widely accepted systems because of, again, problems through catastrophic events, through air quality and water quality issues. But the agroforestry farming systems are still not as widely accepted because we are still in a society where people don't like changes. So that's where we are. But again, it's easier said than done. It requires careful planning to avoid competition of light, air, water, and nutrients. We have to choose the different species carefully. The initial setup involves more labor and cost, and the knowledge. There is intensive knowledge requirement for farmers and practitioners in the beginning. But again, agroforestry is more about mimicking nature. So if you look at savanna and how agroforestry systems operate in many parts of the world, that's what natural ecosystems is what we find. Benefits of agroforestry systems, I'll not spend too much time here, but many of these are related to how soil systems will work better with carbon sequestration, microclimate regulation. But what is more important is that diversity boost, having that wildlife habitat, pollination, and all of those benefits that we get from a biodiverse system. So these are some of the studies that we did in various landscape, comparing agroforestry, row crops, and silvopasture systems. And we saw that agroforestry systems were performing better in terms of microbial diversity, as well as in terms of bacterial diversity. We also saw similar effects in soil enzyme activities. So this is some of the examples of agroforestry systems, how we can incorporate in various landscapes. So I understand that not every situation will work for every farmer or landowner, but depending on landscape, so whether you can incorporate a riparian buffer, which is in the extreme right corner, bottom right corner, or whether you want to incorporate a silvopasture, as you can see the goats grazing, or alley cropping, or forest farming, and you can see wildlife, which you will have a habitat by having trees and other landscape habitats in the system. again, there are challenges in incorporating these management practices. So again, I mentioned initial setup cost and limited knowledge, resistance to change in terms of traditional farming. People don't like to change the way they do things. So it will need some sort of incentives. Obviously, there is a path forward. So we have to maintain soil health, and having trees in the landscape will help with carbon sequestration, nutrient cycling. Having that deeper layer of the soil come into the picture, having that nutrient recycling will definitely help. And the take-home message, soil health is central to achieving resilient and productive agricultural systems. Every management decision, whether it's tillage, rotation, input size, grazing, it has ripple effects on soil biology. So we have to look at a systems approach, and agroforestry is a great way to harmonize agriculture. And I would like to end with this. All of us know Aldo Leopold, and we abuse land because we regard it as a commodity belonging to us. When we see land as a community to which we belong, we may begin to use it with love and respect. Thank you. And I guess I went a little bit over time.
Okay, thank you so much. Looks like we do have one question in the chat. Maybe you can grab that as we go ahead and turn to a fellow Buckeye of mine, Alan Sundermeyer. Alan, you're up.
Okay, thank you. I'm located here in the Midwest, Northwest Ohio. I want to give credit to our United States Department of Agriculture funding through the Sustainable Ag Research and Education grant that I received, a partnership grant. So this helped with the funding. The Conservation Action Project, which I'm the coordinator, the website capohio.org. We're working here in the northwest area of Ohio with the Maumee River watershed, looking at innovative agricultural research and projects and soil health. So I'm going to share some of the tests that we've done and some of the specific lab reports that are included in that. So biological testing, soil health testing, plant testing, you can give it a lot of names. This is a list of the ones that we're going to discuss here today. I'll get in more detail with that. So we look at different labs. Here in Ohio, we have Brookside Lab, and Conservation Action Project has also looked at Region Lab, located in Nebraska with Lance Gunderson. But like Sean mentioned, there's a lot of different commercial labs and university labs. So I'm not saying anyone's better than the other. We just decided on this lab. So some of the reports will show these labs, analytics here. We look at cover crops and we try to see what the benefit of a cover crop is. And by doing an analysis of that forage, by cutting off the above ground biomass, sending it to a lab, getting the dry matter content, and then you can see the value of that cover crop. The trouble is, when are these nutrients going to be available to the next growing crop? What percentage of that available nutrients should we be using? And when is that nutrient going to be available? So that's part of what we're looking at now is multi-times of looking at the cover crop harvest and analyzing it at different growth stages. So basically, the longer you can let a cover crop grow, the more nutrients and more dry matter it should produce and the more benefit to soil health. We talked about the Haney test, and this is just one example of one of the reports. You can see there's like soil respiration, microbial active carbon, carbon nitrogen ratio, and it gives a soil health score. And it compares here then the nitrogen comparison of what a traditional soil test might have versus what the Haney test. And the Haney test with the nitrogen has different extracts. When we look at the water extract and the H3A extract, that gives us a estimate of what the soil has capable of in producing nitrogen. So when you look at a standard chemical soil analysis, it doesn't go into near the detail that you might see here in the Haney test. So how do we use this? Well, with the Conservation Action Project, we look for an average location of soil type in a field, identify that, and then compare from one year to the next. Maybe as we had cover crops, we reduce tillage, we look at crop rotation, and we see that specific spot, how it changes over time. And that gives us an indication if we're actually improving things or if we need to maybe make more management changes to make things even better. So looking at the different reactants here with the Haney test, this is why, when we look at a standard chemical test, it basically takes the chemistry of what is available there in the soil, where a Haney test is looking at what the plant has available. And that's through the water solvent or the H3A solvent. So you can't really compare numbers from a standard chemical test to a Haney test. They're not really looking at the same extracts. The malic 3 is a lot different than the water or the H3A extract. So we have to be careful about making these comparisons. When we look at the PSNT, which is the pre-sidrous nitrate test, basically it's the nitrate levels in the soil. And this is using, again, a different extract than what the Haney looks at for the nitrogen in the soil. But we see a very good correlation between the two. So even though we don't have the exact same numbers, we can see the same indication that as the soil is tested at different levels of nitrate availability, the Haney and the PSNT tests come out very similar in those trends. So one can be related to the other. It's really hard to make correlations between these different tests and to say which one is more accurate than the other. Well, it kind of depends how much money you want to spend and if you want to do the tests locally or send it off to a lab. So that makes some of the difference here. The Solvita soil test is looking at soil respiration. So what we can do is basically take a bulb planter that you can screw into the soil and not disturb the soil, remove that and put it in these cups and let it sit for 24 hours. And you put like a diode probe into that cup, and then it changes color. And it kind of reflects the different colors here on this chart. So again, it's not the most accurate, but it's a simple, easy test that you can do yourself, and you can decide, where in the soil you want to do it, and it gives you an indication of what the soil respiration is. Now that can be compared to like the Haney test, which gives us a soil respiration where they actually dry the soil, re-wet it, and get a much more accurate indication of that. But the two come up with the same result. It's just a different level of accuracy here is what's involved. So this is a PLFA test, which we talked about before, which is a phospholipid fatty acid test. Basically, this is looking at the living microbial biomass in the soil. So here you can see it's a total biomass value, and it gives you very specifically like the total bacteria, the total fungi, and what's made up of some of this, the protozoa. So, it's very detailed, and it can then tell you if you're trying to get a more healthier soil, we like to see the total fungi and those ingredients improve versus the bacteria. The bacteria base is usually not as beneficial for soil health. We want to see that fungi improving more if we can. So the PLFA test gives us a very specific indication of this, and those numbers, can be then compared from year to year or different field to different field. Next is the bee crop test. And this is kind of like looking at the DNA of the soil, one way I can look at it. It's kind of expensive. It's probably over $200. We can look at the genomics, the microbial functions, and it gives you readouts that are color coded, so you can make some general recommendations that way. This kind of gives us different categories of soil quality, plant health, nutrition. All these are very detailed and very specific, and it takes a little time. It might take a month or more to get the results back. I think a lot of this is sent to California. So when we look at row crop testing, you might do this once to get a baseline. It can also help predict if you're going to have any soil-borne disease problems, or also the beneficials, and give you a rating of that. So if you're looking for details, this is one of the tests that are newer. We don't have a lot with the row crop test results yet. It's probably more on the higher value crops like vineyards and such that you might see more benefit from this. But again, you know, in research, if you're looking for very specific things that are going on in your soil, almost like I said, the DNA of the soil, the bee crop test is one of these are available. The total nutrient digestion is another measure of the soil, and it gives you what is totally available. So it's more caustic. It really strips out all the nutrients that are available in the soil. So it's not the same as a malic 3 extract. So you can see, you know, the potential of the soil. We say, you know, a lot of the phosphorus and potassium is tied up, especially in clay soils. And you can then start out with this value. And then over time, you can see if you're actually utilizing this store of nutrients that the total nutrient digestion analysis report can give you. So permanganate oxidizable carbon, or epoxy test, is looking at active organic matter. And this is a fairly new test. Basically, it's looking at a pink solution, as you can then look at the different colors that this then compares it to. So the clearer, the better the soil is, which you look at with the epoxy test. So the more pink it is, it's not as good that way. So Prolific Earth Sciences, I think they're based in the state of Washington, has developed a field test, which the Conservation Action Project, we've probably looked at over 100 of these now. And I think it's a very useful, not quite as accurate again, but it tells us basically 3 things. The microbial biomass, which is expressed here. In this case, it's 236. And then it gives us a percentage of fungal and bacterial content in the soil sample. So you can do this for about $10 or $15 a sample once you buy the initial kit, which is about $150, and it takes only about 20 minutes. So I think this is useful as you compare, again, different management practices in your soil. We've compared this to someone using a lot of manure and looking at cover crops, very healthy soils. His microbial biomass numbers were probably in the 600s to 800s. We compare this to tilled soil, no cover crops, a bad crop rotation, and we'd get 100 as far as microbial biomass. And in those kind of soils, we might only have 1 or 2% fungal, where with a very healthy soil managed well, we could get actually 50% or more fungal content. So this, to me, gives us a quick indicator. Am I doing something right with my soils? And I can do this myself, and it's very economical. And I can maybe compare that to PLFA or a Haney test or a bee crop test, and that would then give me a lot more very detailed information and see if that matches up to what this microbiometer test can tell me. So again, different levels of accuracy, different levels of expense that we may want to put into our testing, and this gives us that choice here. Along the same lines, Soil One is a kit that was helped develop by Dr. Islam, and this looks at basically the epoxy test, and we can then have other interpretations from the active carbon, the active nitrogen, microbial biomass, aggregate stability, So it's a simple test. You can do it yourself. You just put some soil and put some extract with that, shake it up for 2 minutes, let it set for a few minutes, and then again, compare the color content of the pink solution. So it's very simple, it's inexpensive, again, not quite as accurate. So the epoxy test in laboratory will give us a very exact number, and they're using maybe a little more sophisticated process, but this Soil 1 test is something you can do yourself and get a very good approximation versus what a soil lab might give you. Looking at plant tissue testing, the New Age lab that's out of Michigan, it does a sap analysis. And this is, again, a fairly newer test, and it looks at the xylem and the phloem in the plant tissue, where a standard tissue test, like in corn, we would look at maybe the leaf by the ear of the corn, and that would be ground up, and that would give us a nutrient analysis of that leaf. The sap test looks at the difference between the two parts here of a leaf anatomy. So what we're doing with the sap test is comparing the lower part of the plant to the upper part of the plant. And we can do this at different growth stages. These are fully developed leaves that are still functioning. So in the bottom of the corn plant, we don't want something that's yellow or dying. So it might be 3 or 4 leaves up. And then at the top of the corn plant, it has to be fully developed. And what we'll do then is compare the nutrient content of the bottom versus the top of the plant. So what do we want to have? We want to have more nutrients at the top of the plant because that's where most of the photosynthesis is occurring, most of the energy production is occurring. So we want to see that flow of nutrients going from the bottom to the top of the plant. And this is what one of those reports looks like. So basically you have a light green and a dark green, and when you compare that, you want to see that the top of the plant, which is the dark green, has more nutrients versus the bottom of the plant, which is light green. And we get into very specifics as far as parts per million on the analysis. So at a glance, you can see where you might have problems. If you do this soon enough in the growing year, you can maybe add, some foliar sprays to help get that back in balance. Or in the future, you might adjust your cropping plans or your fertility applications to get that in better balance. But this is, again, another very specific measure of what the plant nutrient content is at that specific time, and that can help you then make better management decisions on your cropping and your nutrient applications. Another test at the end of the season in corn we can do is check for the nitrate level in the corn stalk. We take the bottom of the corn stalk and have that analyzed, and then we can see the nitrate percentage in parts per million. The sufficiency range is kind of wide, like from 700 to 2,000. So in this example, we have one that is probably over applied of nutrients at 2,590 is above the sufficiency range, where the other example, 890, was below the sufficiency or very close to it. So what does this tell you? Well, it's at the end of the season, so you can't really do anything about the crop at this point. But when you do this and you compare maybe different nitrogen rates on the corn that was applied, and you can see consistently that you have high nitrate levels in your corn stalk, you probably could then adjust in the future and reduce that somewhat. And conversely, if your corn stalk nitrate is very low, you may have sacrificed some yield by not having enough nitrogen. Now we got to be careful, if you get into drought or different growing conditions, this may not be quite as accurate. So this would be on a very healthy corn stalk is when we do this, and usually about at black layer time as the corn stalk is mature. So as you look at, all these different tests, why are we doing soil health testing? We want to maybe quantify what we think is a good best management practice. We want to quantify, are we doing our no-till long enough? Are we doing our crop rotations in a certain way? Are we comparing different soil types? Are we looking between what is available to us to understand if we're looking at biologicals now, adding to the soil or to the plant, into the seed. Are any of these effects improving my soil health? And then are those effects improving my bottom line of profitability? A lot of these biologicals and other inputs are very costly. So we want to make sure that the money we're spending is spent wisely, and our soil health testing can allow us to do that very, very accurately. Or if you want to save money and do it yourself, you don't have to spend a lot of money and get a good handle on it by getting, an approximation of that. So the level of accuracy is important. How much money you want to spend is important. How many tests you want to take in a field, of course, that more cost. So a lot of these things, it's a field by field basis and what your goals are in this testing. So with that, we're going to continue this here in Northwest Ohio, doing these comparisons between no-till with without cover crops, different soil types, and we're trying to help farmers realize that some of their best management practices truly are changing their soil and improving their soil health. So with that, I thank you, and I'll get us back on time.
Well, thanks so much, Alan, for that presentation and for being mindful of the time. And we are exactly right back on schedule. We're going to go ahead and turn it over to my friend Arif Rahman, and he's got a fun presentation for us talking about artificial intelligence. That's a hot topic today. So, Arif, take it away.
Yeah, thank you. Okay, good morning, everyone. My name is Arif Rahman. I'm working as a scientist at the Ohio State University South Center, Soil, Water and Bioenergy Resources. I'm working with Dr. Rafiq Islam. He's one of my mentor for soil chemistry and soil health. Let's begin with artificial intelligence.
Hello, everyone. Soil health is the foundation of sustainable farming, but understanding it isn't always simple. Traditional soil testing is time-consuming and often leaves farmers wondering, what do I do next? That's where artificial intelligence comes in. By analyzing key soil indicators like pH, organic carbon, nutrients, and even color or redox potential, artificial intelligence can instantly tell us how healthy the soil is and what it needs. Using smart apps and models, farmers can now receive real-time soil quality scores, along with custom recommendations for crops, fertilizers, compost, and cover crops tailored to their location and goals. Artificial intelligence helps make soil data simple, actionable, and affordable, bringing precision agriculture to every field. Let's use artificial intelligence not just to grow crops, but to grow smarter, healthier soil. Thank you.
Okay, let's begin. Soil quality calculation is not easy because soil has physical indicators, chemical indicators, and biological indicators. There are lots of indicators. To test one sample, you have to go through all kind of indicator parameter you have to analyze. My previous fellows already explained everything, each and every parameters. It's not easy to analyze. You have to have a qualified laboratory, as well as you have to spend money. For farmers, doesn't have that much money to spend every time. So physical indicators like water infiltration, soil texture, porosity, soil color, bulk density, soil structure, water holding capacity, chemical indicators such as cation exchange capacity, nutrient levels, electrical conductivity, organic matter content, soil pH, and base saturation. And what about the biological indicators? Like soil structure, soil biodiversity, bacteria, fungi, soil respiration, enzyme activity, soil microbial biomass, and mineralizable nitrogen. It's a lot of parameters you have to analyze. So to get rid of this kind of tedious work, but how we can avoid is there is no way if you actually want to get a really soil quality index on soil health. For example, this is the conventional calculator. This is the conventional soil quality calculator. where you have to give input sand percentage, silt percentage, clay percentage, bulk density, available water capacity, aggregate stability, soil pH, EC, CHC, organic matter, nitrate, available phosphorus, exchangeable potassium. So you have to have those kind of parameters, and you have to go through analysis, and you have to find some qualified laboratory. The right hand side, this is actually soil health map, Ohio. We have all developed through our data, online data, and Ohio Agricultural Department, they provided some data. So if you analyze all kinds of analysis, if you look at the physical properties, chemical properties, biological properties, so if you go for each properties analysis, you have to spend a lot of money. Altogether, average cost $140. So who is going to pay those money? Farmer doesn't want this.
It does.
If you have those data, and if you add, please, you have to add all kind of parameter and soil quality calculator. If you put all those values, then we will be able to analyze the soil quality index. That means soil health values. Recently, we have developed this comprehensive soil health dashboard. After putting all those values, you just need to click. One click will give everything.
Hello everyone. I'd like to walk you through the comprehensive soil health calculator we've developed. This app allows you to evaluate soil health by inputting 3 key categories of data: physical, chemical, and biological soil properties. For this demonstration, we've pre-filled the form with average soil data from Wood County, Ohio, a representative agricultural region. Once the data is entered, you simply click on Analyze Soil Health. Within seconds, the app calculates and displays a Soil Quality Index, SQI, score out of 100, giving you a clear sense of your overall soil health status. The app also identifies your soil texture. In this case, it's classified as loam, based on the sand, silt, and clay percentages. On the right-hand side, you'll see a graph showing the individual SQI scores for each parameter. This helps you visually identify which factors are strong and which need improvement. Additionally, there's a comparison chart that shows how your soil's current values align with ideal benchmark values. This makes it easy to spot deficiencies or excesses. Finally, the app generates customized soil management recommendations. These suggestions are tailored to your data and include soil amendments, cover crops, fertilizer strategies, and other practices. You can also correlate each recommendation with the individual parameter scores shown beside it, helping you connect specific issues to targeted solutions. In summary, this tool offers a comprehensive, data-driven interpretation of soil health, along with clear visualizations and actionable advice to guide better land management.
Since we have already mentioned the number of parameters we need to put into the calculator, to get rid of this kind of tedious work, we have already find out 3 core indicators, which are very important. One is physical, one is chemical, one is biological indicators. But you have to pay money. The cost of analysis around $75 if you find some good quality laboratory. So based on the 3 core parameters, we have also developed a calculator. Let's check it
Hello,
out.
friends. We are excited to introduce our Soil Health app, a powerful tool that not only helps you assess your soil health, but also allows you to compare it with your county's average soil health data. This means you can better understand how your soil conditions stack up against others in your area and make informed decisions to improve productivity. How does it work? To determine your soil health, you only need to input 3 key soil parameters. Soil physical parameter, mean weight diameter MWD of soil aggregates. Soil biological parameter, soil microbial biomass. Soil chemical parameter, soil organic carbon. Step-by-step guide. Enter the soil organic carbon value. Input the soil microbial biomass value. Add the mean weight diameter of soil aggregates. Click Calculate Soil Health. Instantly, the calculator will generate your soil health score. Compare your soil health with county data. Want to see how your soil compares to the average soil health in your county? It's simple. Select your county from the drop-down menu, 88 counties available. Click Retrieve County Data. Instantly, you will see county name, soil type, average soil health score, soil organic carbon percent, soil microbial biomass, mean weight diameter of soil aggregates, soil pH and organic matter percent, soil texture and drainage condition, irrigation recommendations, average rainfall in inches, temperature during the cropping season, average corn, soybean, and wheat yield in your county, cover crops commonly used in your county. That's a wealth of information, just one click away. Hello, friend.
So recently, Dr. Rafiq Islam invented soil quality test kit, and it is in the market. If you check out the price, like see, $45 per kit box. More than 20 samples you can analyze. So instantly you will get the soil health result. Cost of analysis per sample is $2 only. If you mix the soil with the reagent, it will give the following colors. So based on the colors, you will get the soil health, like poor, fair, good, or excellent. But this is completely manual. We try to make it more digital way. So we'll take those reagent color and solution after analysis. Then we will measure how. We have 3 options. Let's check
Hello,
it.
this is a soil quality assessment calculator with 3 available options. Camera image capture, solution color measurement, input of measured solution absorbance value. You can use any of these options to analyze soil quality. Camera image capture. If you choose this option, click on it and a camera window will open. Place the solution in front of the camera and click the Capture Image button. After capturing the image, click Run Soil Analysis, and the results will be displayed immediately. Alternatively, you can upload an image from your device and click Run Soil Analysis to obtain the results. Solution Color Measurement. Click on this option if you prefer to analyze soil quality based on color. We provide 4 different color options, which correspond to the reaction between soil and our reagent. If you select a color, such as pink, and click Run Analysis, the results will be displayed immediately. Measured absorbance value. If you have already measured the absorbance at 550 nm wavelength and have the data, select Solution Absorbance, enter the value, and click Run Soil Analysis. The results will be displayed immediately.
So this is the easy way. If you have this kit and if you see the color, and immediately you can use this app. So it will give soil quality with important soil quality parameter and its associated values.
Hello.
So recently we have also developed AgriGPT, the artificial intelligence integrated app. Let's check it out.
Everyone, and thank you for joining us today. We're excited to introduce you to AgriGPT, our smart, AI-powered agricultural assistant designed specifically to support farmers, educators, and researchers. This app allows you to ask any agriculture-related question, whether it's about soil, fertilizer, crop selection, irrigation, pests, or cover crops, and you'll get an instant answer powered by artificial intelligence. You can simply type your question, and within seconds, AgriGPT provides smart, personalized recommendations. Now, take a look at the top of the app. Here we have an interactive, colorful map of Ohio's soil health. You can zoom in or out, and when you click or tap on any county, it will show the county name, the soil health score out of 100, details like average soil pH, soil classification, and dominant soil texture. The map uses a clear color legend. Red shows poor soil health. Orange indicates fair soil health. Green means excellent soil health. This visual makes it easy to compare your county's soil health to others and spot areas needing improvement. Let's move down to the chat section of the app. Here, you can ask real-life farming questions and receive helpful guidance instantly. For example, my soil pH is 4.8. What should I do before planting corn? AgriGPT will immediately reply with lime application rates and other recommendations to improve soil conditions for corn. How much nitrogen should I apply for tomatoes in loamy soil? You'll get a fertilizer plan tailored to your soil type and crop needs.
So we also developed another carbon sequestration app.
Hello and welcome to our carbon sequestration analysis tool. This tool is designed to help farmers estimate their carbon sequestration rate when using no-till farming with cover crops. By using this tool, you can understand the impact of your farming practices on soil carbon storage, explore financial benefits through carbon credits, and assess long-term sustainability. How to use the carbon sequestration analysis tool. Select your county. Click on the County tab to get the average temperature degrees F and average rainfall inches for your targeted county in Ohio. The results will be displayed instantly, providing weather insights essential for carbon sequestration calculations. Input your soil data. Enter the soil organic carbon percentage for your soil. Add the soil bulk density value. Choose your crop. From the drop-down menu, select one of the 3 available crops. Corn, soybean, wheat. Select your farming practice. Choose between conventional tillage or no tillage from the drop-down menu. If you select no tillage, you will then choose a cover crop from 5 available options. Cereal rye, annual ryegrass, crimson clover, oats, radish. View your carbon sequestration over time. After selecting your cover crop, click the 1 to 5 year sequestration button. At the bottom of the screen, you will see estimated carbon sequestration values for 5 consecutive years. Check your carbon credits.
Let's see our cover crop selection guide.
I'm excited to introduce you to our Cover Crop Selection Guide app. Using this app is very simple. First, you'll see a window where you can select your farming goals. For example, whether you want to improve soil health, fix nitrogen, control erosion, or manage weeds. Next, you can choose which cover crops you would like to rotate into your system. Once you've made your selections, simply hit the button Get Cover Crop Recommendations. Instantly, the app will display a customized list of recommended cover crops. Along with each crop, you'll see key information. Planting months, termination months, seed cost per acre, termination cost per acre. Plus, you'll also receive smart recommendations from our AI agronomist, offering tailored advice based on your selections. If you prefer to listen instead of reading, you can simply hit the button, Speak Recommendation again.
So we have also AgriBot. We have recently leveled up. Let's check it out.
We have developed AgriBot specifically for our farmers, so they can get real-time support anytime through AgriBot, our intelligent assistant. With AgriBot, you can view the Ohio Soil Health Map and interact directly with AgriBot by asking questions about your soil, crops, or land management. Let me show you how it works. If you simply type hello, AgriBot will automatically respond. Hi there. How can I assist you with your farming question today? Now, let's ask a real question. I would like to learn more about soil quality in Pike County. Could you provide some insights? AgriBot will instantly reply. In Pike County, the soil is characterized by thin, eroded Appalachian soils with a soil health rating of 38% and a pH of 5.9. It's important to consider using cover crops like cereal rye and hairy vetch to improve soil quality and fertility. You can ask any farming-related question about soil health, cover crops, fertilizer recommendations, or crop selection, and AgriBot will respond with science-backed, location-specific answers tailored to your needs.
Thank you. That's all.
Thanks everybody for being here today, and for sticking with us through all these presentations here. So today I wanted to come here and talk a little bit about what Syngenta is doing in the soil health space. We've got a global soil health team that is about 2 years old, and so I want to just kind of give some insights on what we've been up to and how we're looking to really partner with farmers and with growers on their soil health journey. So I think the first thing is every time I have a conversation about soil health, the absolute first thing is, people say, well, I don't even know what soil health is. And, similarly, when we were getting our team together, we were posed that same question. Now, I asked AI, of course, it's the new favorite thing that everyone likes to talk about. What is soil health? And this was the image it generated, which anyone who's ever seen a tractor or combine knows that this might leave a little bit to be desired here. But for Syngenta, for our team, what we think of about soil health is we think that soil health is the continuous science-based pursuit of products and practices to harness the potential of soils to optimize grower outcomes. And so I want to emphasize a couple different things in that definition that we put together because it's really important to us. So the first thing is that it's science-based. That is to say that we want to make sure that we are following the data and that we are using that data to produce actionable insights. We want to be reliable partners for growers, which leads me to the second thing that I want to emphasize here is that we don't think of soil health as a prescription or just as a destination, but rather it's a pursuit. There's always more that you can do to improve your soil health to get it just a little bit better. And so we want to be a partner on that journey. And the last thing is that we're really looking to harness that potential of soil. We've had the green revolution and there are so many technological advances. And, those things like hybrids, germplasm, pesticides, herbicides, and fertilizers that have all helped to increase yields. But soils have kind of gone neglected. They've been thought of as a growing media simply. And I think only recently have we started to have an appreciation for what the soil can provide. And we've heard a lot of presentations today about some of the science and underpinning that. Now, for me, I'm really interested in measuring soil health. This is something that my team does. And I think the first kind of question that I always get is why should we measure soil health at all? And the short answer is that we have a lot of uncertainty in our soil health outcomes. Something that is often presented, right, is people will say, well, okay, if you do these soil health practices, then you will necessarily get these improvements in yield, in water availability, in nutrient use. They make that path seem like it's very short. And if you do this practice, then this will happen. But in the peer-reviewed literature, we see that that doesn't always happen, as well as in people's experience. And this is a great summary. It's a meta-analysis of all the peer-reviewed literature and what happens when you convert to no-till. And so you can see that up here, these are the kind of variable importance, the relative importance of certain things. And what you can see here is that first, the most important factor varies from crop to crop. So for example, residue management is really important in corn, but it's less important in soybeans. Similarly, climate might be really important here, but even more important in soybeans. And so really what we're trying to do is there's so much complexity in soil health, and you've heard some of that discussed here earlier, that we really want to take all of that complexity and really distill it down into something simple and that a grower can make a decision and have a lot of confidence in that decision. And so this is really where we want to position soil health assessment. And that is to say, we want to use soil health assessment to increase our confidence that the adoption of all of those practices that Sugata was talking about earlier will lead to those increases in productivity, profitability, and efficiency, right? So that's really how we're trying to use this, is we're trying to use it as part of a decision-making process. That is to say, you adopt some soil health practice in the hopes that it will pay off for some sort of target outcome. And soil health is really as an intermediary to let you know if you're on the right track. We know that there's no one size fits all for those practices. And there's a lot of it depends. Well, soil health assessment is supposed to help change some of that it depends into, here's what's happening. So how can we measure soil health? Well, again, here's what AI thinks. They think that there's someone in a lab coat, also in a field, apparently half buried in the soil and using what looks like a rubber stamp to measure soil. So we're looking to improve a little bit on AI and show you a little bit of what Syngenta thinks about how we can measure soil health. Now, this is something that my team really wanted to tackle. So one of the first things we did is we wanted to use that peer-reviewed literature to understand, do some of the most common soil health indicators actually have a relationship with yield, right? And so what we did is we went through a bunch of peer-reviewed literature and we found here's a list of potential soil health indicators. Now on the left here, we have what we consider our benchmark. We all know that soil organic matter is a good thing for productivity, right? Same for soil organic carbon. They're somewhat interchangeable. But what we really wanted to understand is, what about some of these other indicators that are kind of emerging? How do those relate to productivity? And you can see some of these listed here. And I think, for me, the important things to bring out here are, one, some of these are only measured on field fresh soil. That is to say, you take that soil sample and you need to keep it refrigerated and keep it from drying out in order to get a reliable measurement from it. So these are things like microbial biomass or dehydrogenase activity. And this is a very specific enzyme that's really a proxy for microbial metabolic activity. Then the ones on air-dried soil, these are perhaps a little bit more familiar, and some of them have been talked about today. So the first one is this permanganate oxidizable compounds. So these are microbially processed polyphenolics, things like lignin, big molecules. Then we have this soil protein measurement, or autoclave citrate extractable. So these are things that are contributing to soil structure and are a pool of organic nitrogen. Then we have respiration, which has been talked about a little bit here, and then aggregate stability as well. So that data set, we screened over 14,000 peer-reviewed studies, and of those, only 65 of them had a soil health indicator and yield, showing exactly how difficult it is for sometimes for us to take these soil health measurements and actually convert them into a decision and to be predictive of something that farmers care about. So here you can kind of see the distribution of both how many observations we have of each, as well as kind of the location. And what we did is from each of those, we took the correlation coefficient from each of those studies. So for example, what's the correlation between soil organic carbon and yield? And yield could have been whatever crop was the one of interest. So we put those together and we want to kind of get a good sense of which ones are performing well and which ones aren't. So here on the right, you can see the average correlation in the box here, and you can kind of see the range of correlations that we found. So just maybe as a point of reference here to orient you, a correlation of about 0.5 is the approximate relationship between adding nitrogen fertilizer and yield. So that maybe helps contextualize some of these correlations here. And you can see first is that most of these, almost all of them, have a positive relationship with yield. Next is that correlation was greater than 0 in almost all of these. You can see respiration, so organic matter, all of these have generally greater than 0 correlation. But there was a lot of variability across studies. So for example, here in microbial biomass carbon, you can see it ranges from about -0.3 all the way up through almost +0.1. So a huge degree of variability there in those correlations. The indicators that were the most strongly linked to yield were the ones that are kind of biological indicators. So this dehydrogenase activity and microbial biomass carbon. Interestingly, soil organic carbon is also outperforming soil organic matter, which is what you would get on a standard fertility test. But, a good start. But the thing is, soil organic matter, specifically loss on ignition, is really inexpensive to measure. You can get it for a couple bucks. It's really part of most routine soil fertility tests. And so to us, that was kind of the indicator to beat. That is, we want to perform better than that. We want to know that we are getting additional usable information over that. So what we wanted to do is we wanted to understand which ones are consistently outperforming. So here, what we did is we took all of those studies where we have soil organic matter and some other indicator, and we compared those correlations and we subtracted one from the other. So here you can see anything to the right of this line here means that that indicator performed better than soil organic matter in a head-to-head on a specific set of fields. And anything to the left here, it performs worse than soil organic matter. You can see that this soil proteins pool, it consistently overperforms soil organic matter, and same for the permanganate oxidizable compounds here. But what you can see is that some of these other more direct microbial measurements, like microbial biomass carbon, they don't offer much additional predictive power over soil organic matter. Similarly, respiration is offering not much predictive power, and aggregate stability actually is performing worse than soil organic matter, significantly worse. Unfortunately, we couldn't compare dehydrogenase activity, which was the other enzyme activity, to organic matter, simply because we didn't have enough observation. So I want to remind ourselves of a really important saying, which that correlation is not causation. And this is a great example of that. This is the correlation between per capita consumption of cheese in the US and the number of people who died by getting tangled in their bed sheets. And you can see that those are really strongly correlated. They have an R of 0.95, much higher than some of our soil health indicators and yield. But the good news is that because correlation is not causation, just because you eat lots of cheese doesn't mean you're going to be tangled in your bed sheets and die. So rest easy. But we want to take that same logic and we want to apply it to our soil health kind of selection. So we want to kind of build on these results here because there are some pretty distinct limitations of this first study. The first one is that because correlation is not causation, we clearly need to build a better model to understand how soil health is causing yield rather than is soil health, say, reflecting changes in yield. The other thing is that our meta-analysis was only looking at one indicator at a time. We weren't really considering how can we best combine these. And the last one is that we want to be able to separate out the act of selecting an indicator and putting in practice its agronomic interpretation. What I mean by that is there are many things that can limit yield, and it's not always soil health. Sometimes you need to add lime, right? And that might not be picked up by some of these more biologically based soil health measurements. And so when we are doing that, we to move from correlation to causation, there's a couple important parts on that measurement step. So the first one is that we want to distinguish inherent versus dynamic soil traits. Something like clay content, while we know it's really important for your soil, it's not something that you can change within your soil, right? It's fixed. And so we want to make sure that those soil health indicators are focusing on that thing that you can change about your soil. The next one is that there's a million indicators, right? Alan was talking about a lot of different tests and indicators, and how to choose which one. Well, that measurements process, we need to make sure that we have a set of criteria that are clearly laid out to allow for that selection of indicators. Then the last one is that those indicators need to be robust across context. That is to say, we need to be able to measure that same dynamic soil trait, whether we are in an arid climate, a temperate climate, or a humid climate. And also, in addition to those comparisons across really kind of disparate fields, we also then need to be able to go within a field and make those comparisons. If you take 10 samples within a field, you should be able to have the same accuracy of measurement within that field as well, really kind of leading to the unlock of soil health and precision agriculture or precision conservation agriculture. Then on the agronomic interpretation side is we want to have this balance of sensitivity and accuracy. We want these indicators to be really sensitive to changes in things that are going out there, but we don't want them to be so sensitive that if you, take your sample in the spring and it dries out slowly versus in the middle of summer and it dries out quickly, we don't want those to overly impact our results. We also want them to be able to give some sort of information back within 1 to 2 years of trying something new. And lastly, the thing that I was previously discussing is that we want them to be leading indicators of change. That is, we want them to change before we see changes in yield. We want them to be predictive of future changes in yield, both in the magnitude and in the direction. So I want to do a quick kind of case study to show, especially on this inherent versus dynamic component, why this is really important. So this is out of Texas, and this was using that Haney soil health test. And these are actually on some of the fields that Rick Haney developed this test on. And so this is really where this test should perform really well. So they went out and they did a really, really high intensity grid sampling. Every 35 meters, they took a soil sample and they ran it for the soil health. Now, what you can see here is that within this field, they had several different soil types. So here is one soil type, here is another soil type, here's another. And so you can start to see that you have a lot of heterogeneity in that field. So what they found is on the left here was their yield map from that year. And so the yield map is the colors here, with green being higher yields, red being lower. So you can start to see that over here, there's a lot of red, right? Similarly, right here, whereas in here, you see a lot of green. And you can see that those yield monitor measurements start to line up pretty well with those soil types, right? They are breaking right along those soil types. Similarly, when you look at the soil health values, with blue being higher and red being lower, you can see that this has generally lower soil health values. Here we have higher soil health values, as well as over here, maybe in the middle. So now when you put those together and you kind of lump all of those soil types together, you get what might be an expected relationship here. That is that as the soil health value goes up, so does yield, right? So that seems good. But we want to be able to go to a new soil type and have confidence that the soil health value will translate into increased yield. And so when we parse it out by soil type, within this field, you can start to see some differences. So here you see a positive relationship. Here in this soil type, it's maybe flat. Here it's negative even, and here it's positive. So what that says to us is that this Haney soil health test is not doing a great job of differentiating between those inherent soil factors and those dynamic. It does not perform equally well across all of these soil types as a predictor of yield. And so then you can start to get into this really circular kind of question where you're starting to wonder, okay, what's the problem here? What's the problem, right? Especially when you're thinking about this from a management perspective. Because sometimes you can take this top approach where you say, we're doing this good management. We're doing no-till, we're doing cover crops, and the indicator went up. So therefore, my soils must be getting healthier. But you can also have this opposite thing where you say, well, the indicator's going up, so therefore this management must be building soil, right? And you can kind of talk yourself into it either way. But when you think about it both ways, you really can't actually effectively tease those different things apart. We can't actually separate those two. Which goes to one of my other points, is that we really need to separate the act of soil health measurement from the agronomic interpretation. So I want to give you a little bit of a case study now of how we're doing this on my team. So this is within the US, a nationwide potato survey. So this was a bunch of on-farm trials. We had about 80 fields. Each of them got intensively grid sampled. We had 20 points per field. And we went out and we were measuring a lot of those soil health metrics that we were talking about before. So organic matter, manganate oxidizable compounds, so protein, et cetera. We were also trying to integrate how pest pressure influences potato productivity. And now, in terms of potato productivity, potatoes are really different from a lot of other crops in that quality really matters a lot. So it's not just the yield quantity, but also the yield quality, which then indirectly leads to revenue. So we were using an economic model to understand revenue as a proxy for quality. So I won't kind of go into the specifics of how we put that soil health index together. It's a lot of statistical details, but I will show you how we were then able to, once we developed that index, how we were then able to move to that agronomic interpretation. So the first thing is that within each of these fields, we estimated an effect size of soil health on yield, both productivity quantity as well as that quality or revenue. And we can see that in about 90% of those fields, that soil health was a positive predictor of both revenue and yield. Now, this is after accounting for things like weather that season, as well as texture. So we were taking out all of those things and making sure to not double count them. But we're seeing a really, 90+% predictive power. And, we were about 92 to 99% certain about those positive effects. Now, if you look at the distribution of these effects, you can see that revenue effects here in red were generally bigger than just increases in yield, right? So you see a little bit of divergence here, and it really shows that you can start to have yield effects that are independent of quality. And so what we did is for each of these fields, we took them then and we said, okay, you have this effect on yield and this effect on revenue. Are they just linearly related? Like how are we, how are these things interacting with one another? So here you can see that those soil health effects, in general, the effect on yield as it goes up, the effect on revenue also goes up. Each of these is a field here. But if you look at the quadrants here, so the top right is you get an increase in yield and an increase in revenue. Best case scenario, right? Here is you actually get a decrease in yield, but an increase in revenue. You're not producing as many potatoes, but the ones you're producing are higher quality, so you make more money. Over here is you can increase yield, but actually decrease your revenue. You're just growing more potatoes, but you're not growing better potatoes. And down here is You're growing less and you're not making as much money. So lose-lose. You can see that most of them are falling here in this top right quadrant. Okay, you're making more money and you're growing more potatoes. But they're not just linearly related to one another. You can see as soil health, as the effect on yield of soil health on yield goes up, that it starts to kind of reach a plateau here. But that plateau doesn't correspond to increases in revenue due to soil health. You can see that the effect on revenue is still increasing, even as the effect of soil health on yield quantity stays the same. So that's a really important finding, is that soil health has both yield quantity and quality effect for these potatoes. So it really, makes me think about other crops where quality is important as well. Now, lastly, the thing is we want to understand, how does this investment in soil health, how does our soil health index actually relate to things like buffering against extreme weather, right? Weather is not a thing that we can control. And so here we want to understand that interaction. So here's a plot of the kind of average effects across all of these fields. And you can see a couple things here. So one is that in the warmer and drier climates, yield is generally, for any given soil health value, tends to be a little bit high. But what you can also see is that the effect of soil health is not the same. That is, the slope is not the same. In the cooler and wetter climates, the slope is somewhat shallow, right? But then as we go up to the warmer and drier climates, the slope is actually bigger, is steeper. So the effect of soil health on yield is greater under more warm and dry conditions, meaning that there's probably some buffering here from those conditions. Soil health is helping you to be resilient under extreme weather for that specific year. And again, we have a really high degree of certainty on this interaction. We're sitting almost 90% certainty on that. So I think I'm just about at time. So I want to give you just a couple take-home points. So the first one is soil health assessments should simplify complexity, not add to it, because we want to use it to make a decision. A lot of the current soil health tests don't really differentiate between those things that are under your control and those things that are not under your control. Then, for our meta-analysis, we found that soil protein and epoxy are better standalone indicators than soil organic matter. And we also found that we can reliably quantify soil health across these really diverse soil. Those changes in soil health that we've quantified can be very consistently related to improvements in yield and profitability. That is, healthy soils give more and better quality potato tubers, leading to increases in revenue. And then those healthier soils also buffer against that extreme weather. Okay, so I think that's the end of my time. I really thank you all for tuning in, and I'm looking forward to answering some questions here if I have left enough time.